Good evening, everybody. Thank you very much for coming on this atrocious fall day with rainfall and everything. Much appreciated. It is my distinct pleasure and honor to introduce today's speaker, Professor Gerald Schatten. He goes by Jerry to those of us who have, I hope, earned the privilege to call him Jerry. He's not only a longtime friend and mentor, but a very, very accomplished scientist and advocate of science. His current, obviously, titles may not be quite represented on this first slide. He's the director of Pittsburgh Development Center. He has a number of directorship titles that will take half of the lecture time if I go through all of them. But I think it's worth summarizing his career that started with the undergraduate and graduate degrees at University of California at Berkeley. He has done postdoctoral work in Germany, in Heidelberg, in the German Cancer Research Center, and a couple of other postdoctoral works. He has had illustrious career in cell biology, reproductive biology, assisted reproductive technologies, developmental biology, and on, and more recently in the last 10 years or so, of course, in regenerative medicine and stem cells. I think it's really important, especially for the younger audience, to underscore that Jerry has made a difference not only as an official mentor of graduate students and postdoctoral trainees in the United States and elsewhere around the world. He has been critical in shaping scientific careers of many, many young people, including me, back in the day when I was young, of course. And his passion for advocating for not only in general for young people in science, especially for underprivileged communities and minorities and women in science, has earned him directorship of a number of courses in the United States that are all financed by National Institute of Health, supporting his work, training new and young generations of scientists. He has obviously published more than anybody could want, over 300 papers, including prestigious journals such as Science and Nature, and a number of other ones, trained well over 50 doctoral students, postdoctoral fellows, and been a mentor probably to hundreds, if not thousands more, through other means. I can continue talking, but I will give him the podium. I hope that you can appreciate the lecture today. Jerry, without further ado, please. Thank you. Thank you, darling.
Thank you. Thank you. Such a pleasure to be here and to participate in the celebration of your 20th anniversary. And I am so overwhelmed by the leadership of Rector Danilo. It probably was delusional 20 years ago, but it's extraordinary what's been built. And the facilities here are truly amazing. And I modified my talk some because I wanted to discuss where I think the University of Novogorica, and I apologize for my Slovenian, might synergize and capitalize on the future in biomedical research. And I will talk a lot about aging. But I want to mention that I have a good friend at Harvard, and his advice is exploit your unfair advantage. He wants everybody to exploit their unfair advantage. And I think here you have amazing unfair advantages. So I don't know I know of rectors in a box, but I don't know of rector's wine in a box. And so I'm very impressed that you already have such an incredible resource here, which you don't export much. In that category, there is a tremendous amount of biomedical research that focuses on alcohol. And perhaps this will dampen wine sales a little bit. I don't think so. But there's a lot of different stem cells here that could be studied in terms of consequences of alcohol in all of our countries. In fact, I think it's up to 3% of the babies in Russia suffer from fetal alcohol syndrome. And clearly none of those babies chose to drink. And so even from a humanitarian perspective, understanding the biomedical aspects of something that also a big community here is working on, I think might bring a synergy. I'm going to talk about aging, and I like this. Birthdays are good for you. The more you have, the longer you will live. And this is an issue for all of our countries. And you'll see that whoops, did I do that? There's global aging. I hope I didn't do that. Whoops. Okay, well, I think that'll go off in a second. The global aging is not only in the West, but it's even in China and other countries. Now, remember, China has had a one-child policy. So for some of the young people, if you're in China, you're going to have to support 2 parents and 4 grandparents. And I'm sure all of your salaries are enormous here, but imagine supporting 6 people all the way through their retirements. And the projections over the next 10, 20 years are really pretty dismal. Here in the EU, and Slovenia is here, so you're not outside of this. In 2060, there is going to be 57% of people over 65 that people 15 to 64 will have to support. So each of you will have to support roughly 1 grandparent. And so again, you can see how your resources are going to be challenged. In fact, they've trying to change the way the EU flag looks because you have such an aging population here. Now, you'll forgive me, Rector, but when the Rector and I need to go into nursing homes, which is not so far away from me, much longer for you, again, you young people will have to pay for us. And there will be an enormous shift of resources from young people to older people unless you want to see us on the streets. And that's one of the reasons that medical research on aging is so important. So this is from about 2 weeks ago. The US Senate is going to give more than $2 billion to the NIH in 2016, and a good percentage of it is going for Alzheimer's research, either $300 million or $350 million. The Senate and the House usually never agree. The Republicans and the Democrats never agree, but they're all worried about Alzheimer's for themselves and for their family. And what's ironic is if you can keep me in a nursing home for maybe 22 months, that's all. If I can continue to take care of myself for 22 months, or if my poor wife, who is a mathematician, takes care of me for an extra 22 months, then our aging demographic problem is solved. So this is not an enormous problem. It's just a problem of slowing the trajectory of Alzheimer's by a finite amount. And I wanted to talk more about your unfair advantages. And for the young people in the audience, Tanya knows this well, we offer courses especially at a place called Woods Hole. The resources here are ideal for an international course or for annual international courses. And so you could become literally the mecca of regenerative medicine, of biomedical training. And I think this is an area that Tanya and your other colleagues have already pioneered and you should build on. Now, one of the creatures that we study is called an axolotl, and these are amazing because you can chop all different pieces off of them and they regenerate. Some of you may have noticed today was rainy. Now, Tanya is a genius, and she figures if it's raining, take them in the cave. And in the cave today, we saw this creature. And this is one of these blind salamanders that you have. What's the name of the cave?
Postojnska jama.
I know it's Yama at the end. Postojnska Yama.
Perfect. Yeah.
This is, again, a unique natural advantage advantage that you have here in Slovenia that you should exploit. And there are other things that you should exploit. Now, this may be our next First Lady.
I hope not.
There are remarkable talents in Slovenia. I don't know about her judgment, I must say. But there are amazing talents here. And the greatest natural resource you have are the brilliant people that you have here. You have an educated, dedicated workforce. And I worry that maybe like the salamander, this could become an endangered species. And so I hope that you can continue to provide the resources so that your young people can continue to thrive. And that maybe when you celebrate in 100 years, I can come back because of the research that you've all done. So I want to explain, and Tanya knows this well, how we're in a unique phase in the biomedical sciences. It comes from a good Arabic world, alchemy, turning lead into gold. And you know that while many people in Europe say, oh, we provided the foundations for modern science, most of it came from Persia and North Africa. And so when you have alchemy and algorithms and alcohol and algebra, I mean, these are all words that come from our friends in the Arabic world. But I want to talk a little bit about Dolly. So when I was a student, we learned that when two gametes come together, you start life in a way, you become a fertilized egg, you become an embryo, a fetus, you're born, you go to school. If you're a scientist, you get a job and maybe a grant or 2 or 3, and then you're dead. And that life is a one-way trajectory from birth to death, and it never reverses. It's only a one-way rectified highway. Yet with Dolly, it was amazing, because Dolly came from a cell from a 7-year-old sheep. And so it's like the fountain of youth. It's like Ponce de Leon. Time was reversed. And that's not supposed to happen in biology. This is heresy. We're all infidels. I mean, it's amazing that in some weird way, time could be reversed. And now scientists like Dolly, I want to just show you the movie. So now scientists are working with embryonic stem cells. So these originally came from human embryos. The embryo is destroyed. The inner cells that are to become the cells that would have made the baby are grown in a dish, and these cells then propagate and they grow forever. They are immortal. And it's strange to think of metaphysical concepts like immortality being discussed in a dish, and yet these cells continue to grow, and in theory, they can be used for a wide variety of diseases. Now, there are a number of different types of these stem cells, but the one I'm going to talk a lot about is induced pluripotent stem cells. So many of our religions have problems with destroying a human embryo. No one can say, when does life begin? There's a joke about that, and where a priest and a minister and a rabbi talk about when life begins. And the priest says, it begins when the sperm and the egg fuse. And the minister says, no, life begins when the brain of the fetus wakes up and that it's conscious in the utero. And the rabbi says, no. Life begins when the kids move out and the dog dies. I mean, so, all of our religions have a different perspective. And so many of us can discuss the religious concerns about destroying a human embryo. But what's amazing is with induced pluripotency, you can take cells from people in their 80s, you can take cells from people in their hundreds, and with a small set of chemical tricks, turn them back into embryonic cells. And this is totally revolutionizing biology and medicine. So let me give you the idea. You start with a patient, and you may have heard how if you have a diabetic patient or a patient with Parkinson's disease or with a bad heart, you might be able to make that person's special heart cells and turn this unhappy face to a happy face. Now, there are lots of reasons this may not work, but also if it does work, it's going to work for maybe Bill Gates and Warren Buffett, old white guys who are really rich, stale, pale males. And it's not going to work for the whole population because it's going to be too expensive. Instead, though, you're seeing lots of pharmaceutical companies extremely excited about working with a battery of patients where we don't even know what causes the disease. So, for example, you can take a battery of patients who have Alzheimer's disease, grow their cells, differentiate them into neurons, and then use high-throughput drug screening to ask what keeps the nerve cells alive longer in the dish. And what's remarkable about this is that you're starting with human cells. So like right now, if you ask me, can we do anything for your mother with Parkinson's? I can say, no problem. We can cure your mom with Parkinson's, as long as she's a mouse. I mean, we can do amazing things with mice, but don't ask us about humans. Right? And what's amazing about this is that here you're starting with the cells from a human. So you don't need to kill a mountain of mice and then find out that the mouse work doesn't extrapolate. And so, for example, this is a different one, but I'll come back to this maybe. So for example, with Alzheimer's disease, they can already take cells from different patients. They can figure out which of the different genetic types the Alzheimer patient is suffering, and then in some cases, they're actually discovering drugs that will cure the cells in a dish. They'll prevent the neurofibrillary tangles from forming in the neurons in the dish, and also they'll prevent the amyloid plaque from developing. And here, if you're religious, you need to say, thank God, inshallah, because God has been good to molecular biologists. It takes 60, 70, 80 years for Alzheimer's to develop in our brain, but in the plastic dish, it develops in 2 or 3 months. There's no reason it should develop that fast unless God loves molecular biologists. I mean, it's a miracle, really, but it does. There's another disease I wanted to speak about, and that is macular degeneration. And again, nobody understood how the eye worked until our friends in the Arabic world discovered it, and they developed something called al-camera that we now call, it's a camera, because they developed the first pinhole cameras. But with macular degeneration, you end up going blind. And now in the UK, in Japan, in the United States, and in Israel, there are studies on blind people where one eye gets injected with cells that will replenish the retina. And this comes from a company that Tanya worked with. What's brilliant about blindness is if I'm blind, I don't have much to lose. You can go ahead and inject one of my eyes because I can't see anyhow. It's also brilliant because you can look in the eye and see how many cells are there. And if you're working with the FDA, they want to know how many cells did you transfer and how many are still surviving. And you can look in the eye and see the retinal pigment epithelium. It's also brilliant because the eye doesn't get the whole immune reaction of many other parts of our body. And it's brilliant because if, God forbid, a tumor grows, you can take the eye out. And furthermore, it's extraordinary because you can measure function. So many of the things that we look at when we're looking at cells in a dish are using indirect markers, so we don't really know if they're functioning. But with the eye, you can just do an eye test and see if the person can see better. So there are lots of different ways in which stem cell transplantation can change medicine for the better, and this is certainly one of them. I did want to give you a little bit of history on Alzheimer's, though. So there was a patient who was getting tremendously paranoid. The husband went to the doctor and said, "My wife is so paranoid, she thinks I'm running around with a neighbor, and I think her brain isn't functioning." And Alzheimer diagnosed her with a dementia, and later on autopsy, they found that there was this unique neurofibrillary tangles. But after she died, the husband ran away with the neighbor lady. So in fact, she was right. He was cheating on her. And, they never tell you that. Okay. Another area stem cells and regeneration can make an incredible differences in the heart. And I want to tell you about what happens in fish. So fish don't have the same complicated heart as we do. It's a more simple heart. But you can cut off a third of the heart, and within a month or two, it will fully regenerate. So you can cut here, and with a human, when there's a heart attack and the heart muscle is damaged, you're going to have to live with that for the rest of your life. In contrast, in many lower animals, you can cut it. It's painful. Be careful. And yet it doesn't form the scar tissue that we form. And so if you can regenerate things in lower animals, and you can regenerate things in a plastic dish, why can't we learn more about the natural regeneration in our own bodies? So for example, if a child loses a finger, if they lose the last digit, they'll regenerate that if they're under 3 years old. So we do have some regenerative capabilities, but often the surgeons will seal the wound and it won't regenerate. But so there's a lot more that we can be doing. There's also a problem as people age. This man isn't hearing the woman very clearly, and he's kind of excited for other purposes. There's a lot of nonsense that you'll see on the web. And so if there's only one thing to take home from my talk, if you have a loved one, don't let them get a stem cell treatment. There is so much nonsense on the web that you can spend your life savings on, and it offers you no medical advantage. This was a company that was advertising your monthly miracle. Guys, don't try this. It won't work for you. Now the company apparently is changing it. There's also a controversy in our field where there's a group that thinks that the ovaries from adult women have stem cells, and that the stem cells can be grown in a dish. Now, most of us don't believe that this is correct, because if this were correct, then we don't understand how menopause takes place. Most of us think that a woman has all of her eggs before she's born. But this notion has been commercialized. Oh, let me tell you about this. So there's an area where women get older, and in 1873, a Harvard professor wrote a book where he found the reason for female sterility. The culprit, he says, Tanya, was women. It diverted energy from the reproductive machinery to the brain, resulting in women with monstrous brains and puny bodies. Now, let me warn you, no man has ever diverted energy to his brain, okay? But there's something terribly unfair about reproductive aging. Men can go on forever, whereas women, from an in vitro fertilization perspective, after about 40, 42, the rates really plummet. And nobody understands, why do the eggs from women after 40 deteriorate? And we know it's the egg because in Italy and also in Southern California, there are clinics that have worked with women in their 60s and in their 70s. And as long as they're getting an egg from a woman in her 20s, their bodies will give birth to babies. I mean, and, there are societal issues about having a baby when you're 70, although men do it, so I don't want to touch this. But there was a clinic that thought maybe we can take the mitochondria, which I know Martina works on, maybe the mitochondria can somehow be supplemented in the egg of an older woman. And there's now a company that's doing this. The company is valued over $150 million on the stock market. Now, what they're doing is they're collecting mitochondria from these egg stem cells. We don't think that those egg stem cells exist, but they have a company worth $150 million to collect the mitochondria from something that probably doesn't exist. And then they're charging infertile couples $25,000 to inject the mitochondria into the eggs. And so far, their pregnancy rates are about the same as if you didn't inject. So you're paying an extra $25,000 to inject mitochondria from something that doesn't exist. They won't permit it to be done in the US. The FDA said no. But in Canada and in the Emirates, they have a long waiting list of people willing to pay $25,000. I love capitalism. I should tell you, there's a clinic in Reno where if you want a baby, they'll guarantee you the sex of the baby. $7,500, and they'll guarantee you it's a boy or a girl. Guaranteed. And if they're wrong, you get half your money back. I mean,
That's a good business model.
you gotta love capitalism. I mean, what? Anyway, okay. I do want to say a word or two about some diseases that we don't know the cause of. And yet, it seems as if autism and schizophrenia are double the rate when the father is over 50 than when the father is much younger. Nobody knows what's causing autism. And I think we have a global epidemic. And I have a brother who has schizophrenia, and I know that's a terrible disease. But autism affects a family from the age of 3 or 4 or 5, and that child lives for a good long time and represents, I don't think, either a happy person or an economic benefit to the society. I don't want to be too harsh here, but if there were a way to understand autism or to have a prenatal test so you could prepare if you have a baby with autism or make a different choice. And so there's something going on with men as they age that seems to have some neuropsychiatric issues. And I think the work that Else has been doing on pain receptors and on other neurons could someday also contribute to some of these neuropsychiatric disorders which cause so much hardship. There are a few theories. I don't think I want to spend too much time on this, except for the guys in the room, every time your heart beats, you're making about 4,000 sperm. And what's strange is that, I won't get too graphic, but your cells if you have a child at 20, your cells went through 150 divisions. But the way that male meiosis works, if you have a child at 50, it's gone through almost 1,000 cell divisions. And there are some people that think it may be the number of cell divisions that are accumulating errors. There's another disease, and this may be the disease that stem cells first help to cure, and that is cancer. Because cancer cells and stem cells are very similar, and they behave in the same way that they're immortal and they grow forever, and they have other similar features. The chromosomes divide irregularly, and eventually you get an uneven number of chromosomes. Some of us are beginning to think that the origin of a cancer is actually a cancer stem cell. And that when you use radiation or surgery or chemotherapy, it's like you're cutting a weed in your lawn. You may be cutting the bulk of the weed, but there's still a root there. And unless you can kill that root, your loved one in 6 months or 3 years or 5 years may not be with us. And I think many of us have experienced the heartbreak of being told, oh, you know, your mom is in remission. And then to hear 3 years later, it's not durable. And we think it's still a cancer stem cell in there. And now there are some clinics that are not only killing off the cancer with the normal cancer therapies, but they're using stem cell drugs. These are drugs that target the end of chromosomes called telomeres. So they're killing the stem cells and also killing the tumor. And they're getting much better results. And this is owned by Gerald, a company in the US. And I was told that if I know the real results, the company has to kill me because they're in clinical trials. But it may be that stem cell insights may eventually really transform the chemotherapeutic outcomes of cancers. And there's another thing that is a real problem with cancers. We now can cure maybe 85-90% of the children with cancer. But often when we cure them, we make them sterile. Now, the difference between how eggs are made and how sperm are made means that the techniques for preserving fertility in these children that have survived cancer have different strategies. But for boys who have survived cancer, there now are some opportunities to preserve their fertility. So if you have a son, not only might you see your son live a good, healthy life, but you might also see your biological grandchildren. And this is an area called oncofertility, trying to preserve fertility after cancer treatments. And there are a couple different ways that it can be done. And there's a clinic in Münster, Germany, that's already doing this with boys. And one way is to take a stem cell and eventually make sperm stem cells, and after the boy is cured, put these stem cells back in and restore his fertility. Now, I worry about this because if you do this and you have one cell that has cancer, are you going to give that man cancer? Nevertheless, there are some methods to do this. Another method is to make sperm in a dish. And our laboratory has contributed to both of these. I'm not going to spend too much time on this, but we've been able to get cells from men and make cells that look very much like sperm in a dish. Now, making artificial gametes is kind of ethically creepy, and I think there are some issues about that. This other method where the oncologist talks to parents and they isolate the boy, and then after the boy graduates from the University of Novogorica and he's already cancer-free, the idea would be that you would transplant those cells back into him and he would be able to have a natural family. But this is an area that is really quite remarkable. We can now make sperm and eggs in a dish, and we were taught in biology you couldn't do that the germ cell line was different from the body cells. And so now, certainly in mice and also in humans, it's moving forward where you can make both eggs and sperm in a dish. And there are a number of implications. So one of these is here with Noah. You can see Noah is talking to all the animals, and it was raining like it was today when Noah was active, and the animals are all depressed because Noah's saying, no, we're just taking tissue samples. We no longer need to bring the whole animals. And most zoos in the world, by the way, are freezing the cells from their animals for the day when they may be able to preserve them that way. Now, another area that I think would be very wise for you to consider growing here is as environmental concerns get bigger and bigger on Earth, and I understand the environmental biology people will be moving here, there is so many things that can be done for environmental toxicology using stem cells. So, all of us are concerned about how environmental toxins are affecting the next generation. There are enormous companies like General Electric that are growing now human stem cells as screens for environmental toxins before they use new compounds on people. And so again, I think there are brilliant synergies that you have in this extraordinary facility here. Well, maybe I can speak for a minute or two to the young people in the audience. And I want to encourage you to have a broad mind in terms of what you're doing with your careers. Now, a few years ago, the Nobel Prize in Chemistry was given out for GFP. GFP is a chemical, a protein that we all use in our cells that labels cells. It comes from a jellyfish. And on the day that it was announced, the graduate student who worked with Shimamura at Woods Hole was asked, well, where are you now? And this is Doug Fraser. He sequenced the GFP. Marty Chaffee noted, he really should have gotten a Nobel. I didn't need the Nobel. He did all the work. They should have given it to him. When the Nobel was announced, he had spent all of his money, he couldn't get any grants, and he was driving a shuttle bus for a Toyota dealership in Alabama. He was making $8.50 an hour. This guy was a nanometer away from a Nobel Prize. And when it was announced, he was driving a shuttle bus. So I urge you to make sure that in your career, you have a plan B. Now, the plan B might be to go into regulatory science. I have who became an attorney, and now he's a patent attorney earning more than all of us together. Have a plan B. Don't only go into such a narrow field that you can't find a position. I want to end with some ethical issues. One of these issues is whether we're rolling dice in the new reproductive technologies that we're using. Some of you may know that Tanya has pioneered all sorts of methods for making genetic modifications in a number of different species. And now there are clinics that are advertising that you can choose the IQ of your baby. So if you want a baby with 130 IQ, that baby will not live in poverty. For men, it won't go to jail. Whereas if your baby has an IQ of 70, 30% have poverty. I like this. 32% of the women have illegitimate children. Excuse me, what about the guys? There are clinics that are now selling this, that you can choose the IQ of your baby. This is utter, complete, total nonsense. You cannot do this. By the way, if your baby has 130 IQ, they can become an attorney, a chemist, an executive, not a physicist, not a biologist, excuse me. This is nonsense, but people are paying money for this. And they're paying money for this in part because the work that we did with Tanya, where we made the first genetically modified monkeys, we did this by using a retroviral insert. So we were able to use the green fluorescent protein that was brought into the cell, and it modified with this little piece of RNA that later got converted into a single strand of DNA and then double strand of DNA. It modified the DNA of the egg, and later it was used for transgenic monkeys. Now people are using this to make monkeys that have Huntington's disease or Parkinson's disease, and they're using this for animal models that might be able to help us. But the foreign DNA goes into the blueprint of the egg, and then later we came and injected the sperm into the egg, and development proceeded. But there are much better methods now than the methods that we used. Here you can see the sperm coming in. I wish I had the theme music from Jaws here. But and then the egg develops and whatnot. There are much better methods that, wait, I'll come back to that. I cut it off. There are much better methods with humans now. And also, there are methods that have been being used in the UK where you can have a baby with 3 parents. You can get the DNA from the nucleus from one woman. You can get the mitochondrial DNA from a second woman. And then you can get the father's genetic material. So already they're testing this in the UK to have 3 genetic parents. Now, some of us worry about whether these babies will be normal or whether it's ethically appropriate, but things like this are moving along. I must say that we teach courses around the world. I love the fact that we've offered courses in Jordan and other places. Some of the methods have been extrapolated for other purposes. And what amazes me is how we see the world through the faith and the traditions that we were brought up in. And so when you look at a slide like this, most of you see one thing. If you show it to children, they see 9 dolphins. So there's a dolphin here, and a dolphin here, and a dolphin. Do any of you see a dolphin? I love you. So it's only people with filthy minds that do not see dolphins. But I show this slide because when we grapple with ethical issues, we all have different lenses that we see. And I think the bioethics or the ethics of what goes on needs to be important. You need to know, though, that Tanya has been in this field of regeneration for years. Many years ago, she was attempting to duplicate one of my central features. You can even see the jellyfish back here. And as you would expect, she's trying to use a nice wine to activate the whole system. Tanya, didn't work. You're still beautiful. I worry that some of our babies may end up as little surprises. I want to end by thanking you all. I love Slovenia and hvala. Is that right? Hvala. Hvala.
Hvala.
Hvala.
Hvala. Yes.
Thank you very much.
Excellent. Excellent. Thank you, darling. Thank you. So while Jerry takes a sip of water or two to catch his breath, we'll obviously entertain questions. I'm just going to sit. They told me we should be staged like this. So anybody off of the top of your head, any questions? Any questions for Jerry?
If you don't ask me a question, I'll ask you a question.
Okay, let's try this. Let's start with your question.
May I ask you, how many of you are postdocs or graduate students? And are there, so for the students in the audience, and maybe I can expand it to others, are there opportunities for you here in Slovenia? Will you get a job? Will you get a position? So you're okay. Okay, good. Good. Otherwise, in Alabama, they need someone to drive at a Toyota dealer.
No, but it is. Yeah, go ahead.
Who's investing more in the stem cell treatment procedure? Pharmaceutical industry, or there is some other branch of industry that's going on?
Yeah, so that's a very good question. Who is investing more in these types of treatments, the pharmaceutical industries or others? So when we first offered our stem cell course, we had a vice president from Merck come to lecture. And he said, now wait a second, so you young women and men are going to cure diabetes with stem cells. So let me think. I get about $4 billion a year on diabetes, and you're going to cure it, and now why do I want to help you? Now, and so the pharmaceutical companies initially were totally cold to this idea. But recently they realized that you can grow human heart cells in a dish, and human brain cells, and human lung cells, and every other human cell in a dish, so that the pharmaceutical companies can test a new heart treatment on human cells in a dish and not have to spend, say, $300 million on clinical trials and then find out it doesn't work. And I think it was just yesterday, the day before, Pfizer had spent literally $300 million on a new heart treatment, and they realized it wasn't effective. And so if they can save that kind of money, now you're seeing pharmaceutical firms putting it in. But at the very early stages in almost all of our countries, it's the basic research that's done by academics that lead to the discoveries before Big Pharma wants to get into it. So it's, the NIH, it's the EU, and what's terrible in our field is our grants globally are being funded only about 6, 7, 8, 9%. There are some countries, Brazil, India, China, are putting a little bit more money in, but for most of us, it's not it's a lottery system, and it's not, one requires a stable institution to carry you through the valleys, and then you go up to the peaks, but something else has to normalize those ups and downs.
And if I can just add, this is really a critical question, developing this breakthrough technology with stem cell therapies, right? If nobody is willing to invest into it, if you can't find somebody who will develop them to a stage where you can treat patients, it's really a risky business. And we have been in stem cell field now. Jamie published this in '96, so we are 20 years out. And stem cell therapies are, like you said, retinitis pigmentosa is one. There was a clinical trial that Gerald was doing on spinal cord injury that did not go all that well. And this transition of pharma into discovering that they can actually use these cells and not lose the market while they're doing it, I think is becoming critical. Just last week, we had a meeting with Boston Scientific and Pfizer, and I asked the same question. I said, if you were to develop technologies for the future, would you pursue non-cellular versus cellular-based platforms? And I was fully expecting them to say, non-cellular platforms, not anymore.
Oh,
they all agreed, yes. They said, no, absolutely strategical investment in cell-based approaches are becoming huge in pharma. And I think that is so important for our field, because for the first time we can see, well, maybe, just maybe, we can really make this as a therapeutic option one day, maybe for drug development.
And this aging explosion, I mean, this is happening, already, and, there's going to be a war between young and old people on the economic imbalances.
Absolutely.
Thank you all very much. It's really been my pleasure.
Thank you, Gerald.